Examples of X-linked traits include hemophilia, color blindness, color blindness, myopia, muscular dystrophy, etc. These are the various diseases. That is example of X-linked trait. Y-linked genes. Genes located on non-homologous region of Y chromosome. Genes located on non-homologous region of Y chromosome are called Y-linked genes. The Y-linked genes are inherited directly from male to male. Y-linked genes are inherited directly from male to male. In man, the Y-linked genes such as in man, the Y-linked genes such as hypertrichosis is responsible for excessive development of hair on pinna of ear. On pinna of ear, development of hair in man is hypertrichosis. It is due to Y-linked genes. This character is transmitted directly from father to son. This character is transmitted directly from father to son. Color blindness. Color blindness is X-linked recessive disorder where person is unable to distinguish between red and green colors as both the colors appear gray. It is caused due to recessive X-linked genes XC, which prevents formation of color sensitive cells that prevents XC X-linked genes prevents formation of color sensitive cells, the cones in the retina of eye. The homozygous recessive females XC and homozygous recessive female XCY are unable to distinguish between red and green colors. The frequency of color blind women is much less than color blind men. The frequency of color blind women is much less than color blind men. Dominant X-linked gene XC is necessary for formation of color sensitive cells in the retina of eye. Thus, genotypes of male and female individuals can be represented in the following table. Sex, male and female, normal XCY, normal capital C, X capital CY is normal, X capital C capital Y normal, colorblind X small c capital Y, colorblind X small c capital Y. In female, normal X capital C, X capital C. In colorblind, X small c, and carrier is the X capital C and X small c. The inheritance of color blindness can be studied in the following two types of marriages. First, marriage between color blind male with normal female will produce normal vision male and female offspring. In F1, the son's normal vision, but daughter will be carrier for the disease. This is the sex-linked inheritance in color blindness. Color blind male crossed with normal female. It means carrier male crossed with normal female. Genotype are X small c capital Y. In normal female, genotype are X capital C, X capital C. Then gametes of male X small c capital Y, X capital C, X capital C in normal female. Then in F1 generation, the product is X capital C, X small c, X capital C, X small c, X capital C, Y, X capital C, Y. It means that first two are the carrier daughters, that is the 50%, and normal sons are 50%. means carrier daughters and normal sons. This proportion is 50-50%. In second example, marriage between carrier female daughter and normal male will produce female offspring with normal vision, but half of them will be carriers for the disease. Half of male offsprings will be normal, while remaining half will be colorblind. In these, parents carrier daughter crossed with normal male. Genotype is in carrier daughter, that is carrier female, X capital C, X small c, and genotype X capital C, capital Y in male, in normal male. Gametes are X capital C, X small c in carrier daughter. In normal male, X capital C, capital Y. Then the product of F1 generation are X capital C, X C, that is normal female. Then in X small c, X capital C, capital Y, normal male, that is 25%. X capital C, X small c, that is carrier female. And in last, X small c, capital Y, that is color blind male. It means that in the product of F1 generation, that all four products are present in produced in 25%. Normal female 25%, normal male 25%, carrier female 25%, color blind male 25%. From the above example, it is clear that the X-linked recessive gene for color blindness is inherited from colorblind father to his grandson through his daughter. Through his daughter. This type of inheritance is called criss-cross inheritance. See again, from the above example, it is clear that X-linked recessive gene for color blindness is inherited from colorblind father to his grandson थ्रू हिज डॉटर, म्हणजे वडिलांमधून नातवामध्ये व्हाया मुलीमधून. दिस टाईप ऑफ इनहेरिटन्स इज कॉल्ड ॲज क्रिस क्रॉस इनहेरिटन्स. हेमोफिलिया, दिस इज कॉल्ड ब्लीडर्स डिसीज. हेमोफिलिया is X-linked recessive disorder in which blood fails to clot or coagulates very slowly. The genes for normal clotting are dominant over the recessive genes for hemophilia. The person having recessive gene for hemophilia is deficient in clotting factors 8th or 9th in blood. Even minor injuries cause continuous bleeding. Hence, hemophilia is also called as bleeder's disease. The recessive gene for hemophilia is located on non-homologous region of X chromosome, as there is no corresponding allele on Y chromosome to suppress its expression. There is no corresponding allele on Y chromosome to suppress its expression. So men suffer from this disease. Hemophilia. Hemophilia. Women suffers only when both X chromosomes have recessive genes. The genotype of male and female individuals can be represented in the following table. Sex: male X capital H capital Y normal, hemophilic X small h capital Y. Then in female, normal X capital H X capital H, and hemophilic X small h, and carrier X capital H X small h. Like color blindness, hemophilia also show crisscross inheritance. The inheritance of hemophilia can be studied with the help of following example. Marriage between the hemophilic male and normal female. Parents: hemophilic male crossed with normal female. Genotype are capital X small h capital Y crossed with X capital H X capital H, that is normal female. Gametes are X small h capital Y X capital H X capital H, and the products of F1 generation are X capital H X small h X capital H, X small h, X capital H, capital Y, X capital H, capital Y. It means that first two are the carrier daughters. First two are the carrier daughters and another two are the normal sons. And that percentage is 50-50%. 50% carrier daughters, 50% carrier daughters, and 50% normal sons. Sex linked inheritance in hemophilia. Marriage, two carrier female daughter and normal male. Parents carrier daughter crossed with normal male. Genotype X capital H, X small h. For normal male, X capital H, capital Y. Gametes are X capital H, X small h, X capital H, capital Y. The F1 generation shows X capital H, X capital H, X capital H, capital Y, X capital H, X small h, X small h, capital Y. First is the normal female, X capital H, X capital H. Then second is the normal male, X capital Y. Third is the carrier female, X capital H, X small y, small h. Then last colorblind male, X small h, capital Y. Here 20-25% normal male, normal female, normal male, carrier female, colorblind male are produced in F1 generation. In sex linked inheritance in hemophilia. It also shows like a color blindness criss-cross inheritance from father to grandson via daughter. Sex determination. The mechanism by which sex is established is termed as sex determination. The term sex refers to sexual phenotype. In some species, both male and female reproductive organs are present in same organism. It is described as bisexual or hermaphrodite or monoecious. Hermaphrodite means only one sex is present there. On other hand, some species in which the organism has either male or female reproductive organs is said to be dioecious or unisexual. Humans are dioecious. Humans are dioecious means male and female are separate. Sex determination in human being. The chromosomal mechanism of sex determination in human beings is XX, XY type. In human being, the nucleus of each somatic cell contains 46 chromosomes or 23 pairs of chromosomes, 22 autosomes and only one 23rd pair is the sex chromosome. Out of these, 22 pairs are autosome and one pair of sex chromosomes. Human female has pair of XX homomorphic sex chromosomes while male has XY heteromorphic sex chromosome. XX means same there, so this is homomorphic. XY, this is different, so this is heteromorphic. This genotype of female is equal to 44 autosomes plus XX. Male is equal to 44 autosomes plus XY. During gamete formation in male, the diploid germ cells in testes undergo spermatogenesis to produce two types of haploid sperms. 50% sperms contain 22 autosomes and X chromosomes, while 50% sperms contain 22 autosomes and Y chromosomes. In female, the diploid germ cells in ovaries undergo oogenesis to produce only one type of egg. All eggs contain 22 autosomes and X chromosomes. The human male is heterogametic and female is homogametic. If sperm containing X chromosome fertilizes egg or ovum, then diploid zygote is formed that grows into a female child. In this picture, sex determination is shown in human being. Parental generation, male and female. Meiosis takes place that in male X and Y chromosomes. in female X and X chromosome. Gametes are X and Y. Here in female gametes are XX. Fertilization takes place in F1 generation, that X and X, the sperms are X and Y. Here the XX is formed, that is female, and X pairs with Y, then it form XY, that is male. When X pairs with X, then female is formed, and when X pairs with Y, then male is formed. Here two males and two females are formed there. That determination is 1:1. In this F1 generation, that pairing takes place there, fertilization takes place there, and then two males and two females are produced. Then 1:1 sex ratio is produced there. Sperm containing Y chromosome fertilizes the egg. Sperm containing Y chromosome fertilizes the egg. Then diploid zygote is formed that grows into a male child. This indicates that the sex of a child depends on the type of sperm fertilizing the egg, and hence the father is responsible for determination of sex of child and not the mother. Due to lack of knowledge, women are often blamed for giving birth to female child. समाजामध्ये फार मोठा ब्लेम मुलींच्या वरती, स्त्रियांच्या वरती दिला जातो की एखाद्या विवाहित जोडप्याला जर मुली जन्माला आल्या 3, 4, 5, 6, तर हा दोष त्या स्त्रीला दिला जातो. तर याला शास्त्रीय बेस काही नाही. हा मुलगा किंवा मुलगी होणं हे केवळ पुरुषांच्याच एक्स आणि वायवरती अवलंबून आहे. तेव्हा याच्यासाठी स्त्रीला दोषी धरणं हे समाजाकडून होत असलेला एक अन्याय आहे. Sex determination in birds. In birds, the chromosomal mechanism of sex determination is capital Z, capital W, capital Z type. In this type, females are heterogametic and produce two types of eggs. 50% eggs carry Z chromosomes, while 50% carry eggs. 50% eggs carry capital W chromosomes. Males are homogametic and produce only one type of sperms. Each sperm carries a Z chromosome, so this is the homogametic ZZ. The sex of individual depends on the kind of egg or ova fertilized by the sperm. Parents male crossed with female. Genotype 2n plus capital Z. In female 2n plus capital Z capital W. Gametes n plus Z, n plus Z, n plus Z, n plus W. F1 generation shows 2n plus ZZ, that is male bird. 2n plus capital Z capital W, that is female bird. One male bird and one female bird. This is the sex determination in birds. Sex determination in honeybees. In honeybees, chromosomal mechanism of sex determination in haplodiploid type. In this type, sex of individual is determined by the number of set of chromosomes received. Females are diploid. That is 2n is equal to 32, and males are haploid, that is n is equal to 16. Diploid means 2n is equal to 32 and males are haploid, n is equal to 16. The female produces haploid eggs, n is equal to 16 by meiosis, and male produces haploid sperm, n is equal to 16 by mitosis. Female produces haploid eggs, n is equal to 16 by meiosis, and male produces haploid sperm, n is equal to 16 by mitosis. If the egg is fertilized by sperm, the zygote develops into a diploid female. 2n is equal to 32. And unfertilized egg develops into haploid male, n is equal to 16, drawn by way of parthenogenesis.